So today we are going to start our new chapter, and that is
Animal Kingdom.
Animal Kingdom. Okay. So look at here in front of you, there are n numbers of organisms, right? And
Yes,
they
ma'am.
all are, and they all belongs to, yes, Arti. Is image visible to you?
Yes, ma'am.
Yeah, then please answer it. All these organisms belongs to which group? Monera, Protista, fungi,
Animalia, ma'am.
Animalia. Yes, as you can see, all the animals, they all are having different features, right?
Yes, ma'am.
They are anatomically a little bit similar, but morphologically there are huge differences, right? So in this chapter we are going to learn about the how we classify the animal, the whole kingdom Animalia, into different groups. There are some basic characteristics, okay, on the basis of which we classify the whole kingdom, whole animal world into different groups, okay. So in our last chapter, uh, the second chapter, yes, biological classification, in that we have seen in the planet Earth, all the organism, whole biodiversity is divided into 5 kingdoms, right? Robert H. Whitaker had divided the whole diversity, whole biodiversity into 5 kingdoms, right? Monera, Protista, fungi, plantae, and animalia. We
Animalia.
already discussed about the Monera. Monera are prokaryotic cell, right?
Yes, ma'am.
Protista are first eukaryotic organism, right? Eukaryotic
Yes.
unicellular. Fungi, eukaryotic multicellular. Plantae, completely autotrophic, eukaryotic multicellular. Now we are going to discuss about the last kingdom, that is Animalia.
Animalia.
So kingdom Animalia is quite diverse kingdom, okay. There are n numbers and different types of animals you will find in different ecosystem. So basically on the basis of geography, on the basis of geographical region, the characteristic get differ. Are you getting me? The range of kingdom Animalia is from unicellular to the multicellular. Are you getting me? Just
Okay.
that the range is very high and very diverse, okay. You can find the animals in a deep ocean, in a snowy mountain, even in the poles, in the desert area. So animals, some of the animals can survive in extreme condition, whereas some animals cannot survive in extreme condition. So how they can survive in extreme condition and why they are not able to survive in extreme condition? This is only because of their different structure, different anatomy, different features, right? So let's see the general characteristic of animal kingdom. They all are multicellular, right? They
Yes,
all
ma'am.
are eukaryotic, yes, and they are having heterotrophic
Heterotrophic.
mode of nutrition. Yes, they
Yes, ma'am.
either they are directly or indirectly, but they are dependent upon the plants. Yes, they cannot produce their own food, and that's why they are heterotrophic in nature. If you remember from your class 9 syllabus, the cell, structural and functional unit of life. In that session, in that chapter, you must have studied about the structure of cell and on the basis of presence or absence of cell wall. there are two types of cell, plant cells and animal cells, right?
Animals.
So, yes, Aarti.
Yes, ma'am.
Yeah. So, animal kingdom, as we know that each and every organism is made up of cell, right? So, animals are made up of those cells. Animals are made up of those cells which are devoid of cell wall. Yes, the outer layer is plasma membrane and not the cell wall. As they don't have chlorophyll, they don't have chloroplast, and that's why they are heterotrophic. They cannot produce their food. This is the basic common and the basic feature of the animals. Now let's see the characteristics on which, on the basis of that, we are going to classify the whole kingdom, and they are levels of organization, body symmetry, germinal. Germinal means embryonic. Okay, the layer of the cells that develops during the development of embryo. Coelom, body cavity, metamerism, that is segmentation, and notochord. Okay, so let's see the level of organization. We all know that each and every organism is made up of what? Cell. Yeah, and cell is made up of, see, if you can memorize, uh, you will
Cell
find that
is made up of tissues, ma'am.
cell is the basic unit of life. So how the cell is made up of tissue? Hmm. What is the smallest thing in this world, in this universe? The smallest thing.
Cell.
Cell, are you sure?
Atoms.
Atoms. Very good. So atoms are the smallest thing in this world. After that, little bit
Cell.
complex, little bit larger than the atoms are.
A cell, ma'am.
Molecules, right?
Yes, ma'am.
Then, then
Then
then cell. Okay. Cell is made up of what? Air, water. Cell is made up of air. Is it? Can you tell me the components of cell?
vessels, ma'am.
Sorry,
Vessels.
vessels. Cell is made up of what? This is what, this image is related to what?
It's amoeba.
Amoeba. Amoeba is the multicellular or unicellular?
Unicellular.
Unicellular. That means we can consider it as a cell. Yes. So what are the different components of the cell?
Nucleus.
Nucleus. Okay, very good. Then
Membrane.
membrane. Plasma membrane. Okay.
Endoplasmic reticulum.
Endoplasmic reticulum. Very good.
Mitochondria.
Mitochondria, Golgi body, lysosome, ribosome,
Vacuoles.
vacuoles. If it is a plant, then chloroplast. Yes. So
Yes,
these
ma'am.
are what? Organelles. Okay. That means cell is made up of different components: plasma membrane, cytoplasm, nucleus, organelles, right? And these components are made up of what? Protein, sugar, lipid, Plasma membrane is made up of carbohydrate, lipid, right? The lipid or the carbohydrate is made up of what? Atoms, molecules, right?
Yes, ma'am.
So this is the organize level of organization. Is it visible to you, Aarti? Screen is visible to you, Aarti? Hello.
Yes, ma'am.
Is it visible to you?
Levels of organization is there, ma'am.
Uh, and what about this atoms, molecules, and cells?
Yes, ma'am.
Ha. So this is what this is the levels of organization of cell. What we were talking about till here is only about the organization of cell. Okay. Now we are going to learn about the levels of organization in a body. Okay. We are going to talk about whole complete organism. Okay, so levels of organization, there are various levels. Either the organism is made up of cell, either the organism is having clusters of cell tissues, either the organisms are having organs, organ system. Are you getting me?
Yes,
So
ma'am.
on the basis of level of organization, the whole animal kingdom is divided into 5 types. Okay, so the first one is protoplasmic level of organization. Okay, protoplasmic, the word protoplasmic comes from the protoplasm. Okay, what is the meaning of protoplasm? What is protoplasm? Protoplasm is the living content of the cell, and it is made up of nucleus plus plasma membrane or cytoplasm. What will be that? Plasma. Yes, nucleus plus plasma membrane or nucleus plus cytoplasm.
Cytoplasm, ma'am.
Exactly. Nucleus plus cytoplasm. If the whole body of that particular organism is made up of protoplasm, as they are not having prominent, well-developed plasma membrane, they are having a single layer like a membrane, okay, and that's why it is not written as a plasma membrane but the membrane. Okay, so this type of organism is having protoplasmic level of organization, and the example is amoeba. Okay, now the second level is cellular level of organization. Means the whole body of the organism is made up of cells. Okay, they are not making tissues. Though the whole body is having n numbers of cells, but they all are present as a individual cell and not a tissue because they are loosely arranged. Are you getting me?
Yes, ma'am.
Each and every cell is having different function. Okay, and that's why it is cellular level of organization. Is it clear to you? Cellular level, acellular level, or the protoplasmic level of organization?
What
Yes,
is acellular, ma'am?
protoplasmic level of organization is called as acellular. Acellular, because the organism is not having a proper structure of cell. Okay, means the organism is having only one cell, okay, and not the n numbers of cells. Are you getting me?
Yes,
As you
ma'am.
can see in the sponges, porifera, there are n numbers of cells, okay, and they are arranging in a very loose aggregation, okay, but still they are cellular because each and every cell is performing different function, whereas amoeba is having only one cell, and one cell is having n numbers of organelles, and these organelles are actually performing the complete life process for that particular organism, and that's why it is acellular level, because there the performance was perform all the actions was performing by the organelles and not collectively by the cell. Is it clear?
Yes, ma'am.
So the protoplasmic is acellular.
Cellular.
In cellular organization, organisms are made up of n numbers of cells and they are present in a loose aggregation. They are loosely arranged. Okay, they are not compactly arranged and they are not making a tissue. Okay, and some of the cells are performing particular activity together and are performing some cells are performing individually. Clear? And the example was sponge. Now let's see the tissue level of organization. When the similar type of cells, the origin, okay, when the origin of a particular cell is same. So all those kind of cells will come together, make a cluster, okay, and this cluster is called as tissue, okay. If the organism is having tissues and this tissue, okay, particular tissue is performing particular function. In sponge, cells were performing particular function, okay. Here the tissues are performing function. Tissue A will perform one function, tissue B will perform another function, like this, okay. So this type of organization is called as tissue level of organization, and the example is Coelenterata or Neet area. Can you give me the example of Coelenterata? C anemone. Yeah, C anemone is a Coelenterata. It's an example of Coelenterata. Clear? What is tissue? Can you define me what is tissue? Tissue is a group of similar cells. Okay, a group of similar cells that perform specific function is called tissue. Okay, if the organism, okay, if the organism is having when the cells in particular organism, they are grouped together and performing single function, okay, this type of organization is called as tissue level of organization. Now the third level of organization is organ level of organization. Now what is organ? When the similar type of tissues group together, okay, and they make a particular structure which is called organ, and the organ will perform different function, okay, like lung alone will perform different function, liver will alone perform different function, okay. This is called organ level of organization. So example is platyhelminthes. Okay, platyhelminthes means tapeworm, flatworm. Okay,
Yes, ma'am.
next, fourth level of organization is organ system level of organization. Like us, we are having organ systems, right? For respiration, we are having respiratory system. For digestion, we are having digestive system. So what is this organ system? Organ system is a system where different organs group together and collectively they perform specific function. For example, digestion. The whole digestive system is made up of n numbers of organs: intestine, pancreas, stomach, esophagus, right?
Yes, ma'am.
Mouth. Yeah.
Yes,
So
ma'am.
these are what organs, and when they all group together, they make a system. Now collectively what they are performing? Process of digestion.
Yes,
Yes.
yes,
So
ma'am.
this is the organ level of organization. So from Escalminthus, nematodes, to the chordata, all the organisms comes under this level. Clear?
Yes, ma'am.
Now the next basis of classification is body symmetry. So symmetry means what? What is the symmetry?
Dividing, ma'am.
Symmetry means dividing.
Dividing equally.
Exactly, dividing equally. So basically symmetry is the similarity in arrangement of different parts, okay, on the opposite side. If you are arranging different parts on both the sides equally, okay, then this is called what? Symmetry. Yes. So the symmetry, how we decide? Symmetry, actually you are going to study only about the three different types of symmetry. One is asymmetry, okay. Another one is the symmetry, where again we are going to discuss about two different types of symmetry. So all together we are going to discuss about three different types of symmetry. If the organization, okay, if the organization of the body is not a similar, just like amoeba, they are not having a proper body structure, proper shape, okay, they will always show asymmetry. Okay, means you cannot divide the parts of the body in an exact half. Yes, and this type of division, this type of arrangement is called as asymmetry. So some of the sponges, the porifera, what type of symmetry they have? They don't have any symmetry. You cannot cut, you cannot divide the body parts into two equal halves. Okay, so it is asymmetry. Even you cannot divide the body parts of the snail into two equal halves. So this is again an example of asymmetry.
Asymmetry.
Now let's see the symmetry. When you are able to divide the body of the organism into two equal halves, even or even more than that, okay, if you are able to divide the body into four equal halves, three equal halves, okay, or two equal halves, then it is called as symmetry. So on the basis of uh, how many halves you are able to divide is uh, decides the type of symmetry. So first one is the radial symmetry. So what is radial symmetry? If you are able to cut the body of the organism into n numbers of equal halves, okay, and each part is having a similarity, okay, then this type of symmetry is called as radial. So basically what happens, once you pass the plane, okay, from the center axis, okay, and you can from any direction, if you are able to pass the any plane through the central axis and you are able to divide the whole body into equal halves, then it is called as radial symmetry. Clear? This one is similar, this one, this one, this one. Each part is exact copy of other part. Okay? This is called radial symmetry. Whenever you will go through the whole chapter, or as you told me that you already read this chapter, you must have gone through the word biradial symmetry. Biradial symmetry. What is biradial symmetry? What is biradial symmetry? When you divide the body into two equal halves, okay, by vertical plane only. Okay, if you are dividing the body into two equal halves by vertical plane, vertical by vertical plane, okay, then it is called as biradial. Clear? In radial, we are dividing the body from the central axis. Okay. In biradial, we are dividing the body by vertical plane. Okay. So this type of radial symmetry is called as biradial. So C anemone is the example. C anemone. Okay, is the example of biradial. What is the example of radial? Jellyfish. Okay, sponges, cycon. Cycon is the example of biradial. Then hydra, starfish. Hydra, starfish. These are the example of radial. And sea anemone is the example of biradial. So, the organism which shows radial symmetry, they are mostly sessile animals. Sessile means, what is the meaning of sessile? What is the meaning of sessile? Yes, Sarki.
Have jelly like
Sessile means
move with the use of full body, ma'am.
no. If the organism is immobile, means they are fixed in one place. Okay?
Yes, ma'am.
Sessile means immobile. Okay? So, how they will catch the prey? How they will catch the food? They will use radial symmetry. Okay? They will use this radial symmetry to catch the food from all the sides because they cannot move. Okay? Now, the next type of symmetry is bilateral. So, what is the meaning of bilateral? Lateral means longitude. Okay? So, if you pass single plane, one plane longitudinally or laterally, okay, and By this, you are able to divide the body, two equal halves of the body part. Then this type of symmetry is called bilateral. Okay? Are we bilateral? Are we bilateral?
Yes,
Yes.
ma'am.
Hmm.
We are bilateral, ma'am.
Sure?
Yes, ma'am.
Not exactly, because if you will cut the whole body, in one part you will find the pancreas, and in the other part you will find the liver. Okay, only this is the difference. Otherwise, yes, we are. Sure. So the example of bilateral symmetry is, can you give me the example of bilateral symmetry? Invertebrates and all the vertebrates. Okay, if we will remove this point, the pancreas and glands, if we will not be specific with the type of gland, then we are not bilateral. But if we are going with the general statement, the number of structure, then yes, we are bilateral, because at one side we are having pancreas and at one side we are having liver. So by this, the number of glands, number of organs are balanced. Yes. So in terms of that, yes, we are bilateral. The example of bilateral is many of the invertebrates, just like you can see here, the spider and all the vertebrates. Clear? Now the next characteristic is germinal layer.
Ma'am, what is
Sorry. Yes, Arti.
ma'am, what is open type circulatory system, closed type circulatory system?
Open type circulatory system and closed type circulatory system. Now, if the blood, okay, if the fluid is moving or passing the nutrients or anything from one body part to another part in a closed system, in a closed pathway, okay, and that is vessels, okay, or the specific tube, then this type of circulation is called as closed circulation. Okay, and if the blood or any fluid, circulatory fluid, if they are conducting, transferring any nutrients, gases in a open, means organs are bathed in a fluid. Okay, they are not getting separated by the specific tube. They are scattering throughout the body, and all the organs actually bathed in the fluid. Then this type of system is called as open circulation. Clear?
Ma'am, we are a closed type, ma'am.
Yes, because the blood is moving in a specific structure, okay, in a closed channel. The channel is closed, okay, but in the open type, there is nothing like a channel. Why? Because all the organs require nutrients, the oxygen, right? So in open type, all the organs actually bath in the fluid. Okay, so it is open circulation. Clear?
Yes, ma'am.
Shall I continue from this slide?
Yes, ma'am. Yes,
Yeah.
ma'am.
Okay. So the next characteristic is germinal layer. What is germ layer? What is germ layer? In your class 10, you have studied the chapter How Do Organisms Reproduce, right?
Yes,
In
ma'am.
that, you had learned about the sexual reproduction in animals, right? There is a fertilization, fusion of male and female gametes. After that, zygote will get developed. Now the zygote will undergo the process of mitosis, cleavage, yes, and they will form n numbers of cells. First, they, and on the basis of that, Thus, arrangement of cells in an embryo, they undergo the different stages: blastula, gastrula. Yes, remember embryonic development? No. So
Ma'am, I
from
didn't study this. Ma'am.
how the organism reproduce, you didn't?
That chapter we studied, ma'am, but these words are new to me, ma'am.
Okay, no worries. You are going to study all these things in a more depth in class 12. Okay. So for a time being, you just need to know the development of embryo goes through different stages, and these stages, the difference between these stages are nothing but the arrangement of cells, because keep on new cells are getting produced and they are arranging themselves and making a particular structure. So on the basis of that, we are having different stages. Okay. So blastula stage. Blastula stage is the stage where the cells are arranged in such a manner that you can find two layers. The outer layer, okay, trophoblast, and the inner cell mass, means cluster of cells, which is the inner cell mass. So this arrangement, this type of arrangement is the blastula stage, and this structure is called as blastocyst. Okay, now blastocyst will come, move towards the uterus, and now inside the uterus wall, this blastocyst will implant, attached. Then only mother will get to know that she is pregnant. Before that, she won't notify that whether she is pregnant or not. Okay. Once it will attach to the uterus, then only a female will start showing the symptoms, and your body will start giving you the signal or start notifying you that yes, you are pregnant. After that, the next stage is gastrula. The gastrula stage, more cells are getting produced and now they are arranging. Okay, so in gastrula stage, n numbers of cells will start arranging like outer layer, okay, then one more layer, and then inner layer. So outer, middle, and inner. Okay, like earlier it was like a ball, outer layer, and internal layer is a cluster mass. Okay, now it's converted into the three layers. Are you with me? And then there's a small mass like structure. Clear? This is the gastrula. So here the germ cells, the germ epithelium, they differentiate. Okay, they differentiate into three layers. Outermost is the ectoderm. Derm is the dermis. Okay, epithelial, and ecto means outside. So outer dermis is the ectoderm. Okay, then middle one is the mesoderm, and inner one is the endoderm. Derm is the dermis. Okay, so this is what the when the germ cells differentiate and make three different layers. Okay, this arrangement forms germinal layer. So during the gastrulation process or in the gastrular stage, when the germ cells differentiate and make a layers, okay, so whether the body is having two germ layer, whether the body is having three germ layer, on the basis of that we divide the whole animal kingdom. Okay, so in this image you can see this is your outermost, the light color cells. Okay, this layer is ectoderm. This one is the mesoderm. Okay, this one, the red one is the mesoderm, and then internal, the black one is the endoderm. So what is this? This white color, this one, blastocele or blastophore, where the cluster mass of the cells are there. Here, okay, they will again arrange and make a cavity. Okay. So, on the basis of presence or differentiation of germ layer, whether the germ layer is getting differentiated into 2 layers or 3 layers. If the germ cells are getting differentiated into 2 layers, then it will be called as diploblastic. So, diploblastic means the body will have only 2 layers, and that is ectoderm and endoderm. There will be no mesoderm, no middle layer. Okay, this mesoderm is also called as mesoglia. Here it is a mesoglia, which is a mesoglia is what? Non-living layer. Okay, so here in the diploblastic there will be no mesoderm. Clear? And if the organism is showing then diploblastic condition, then such type of organisms are called as diploblastic. Okay, so what will be there between the 2 layer, ectoderm and mesoderm? There will be layer of protein, lipid, and not a cell. Okay, and this layer where the cell is not present but everything is there, the nutrients, the water. Okay, so this non-cellular layer is mesoglia, and that's why it is called as non-living. Okay, cell is the living thing, but other molecules are not living, right? And the examples who belongs to the diploblastic is porifera sponges, coelenterata, and ctenophora. Okay, all the organisms belongs to these classes are these phylums all are diploblastic in nature. If the germ cells get differentiate into 3 layers, proper 3 layers, ectoderm, mesoderm, and endoderm. Okay, then such type of organisms are called as triploblastic. The term diplo and triploblastic. Blast means, sorry, it should be blast. What is the meaning of blast? Blast is a cell. Okay, if the body, if the embryo is having 3 layer of cells, okay, then it will be called as triplo. If the embryo is having 2 layer of cells, then it will be called as diplo. Clear? And the examples from platyhelminthes to the mammals, all the organisms are triploblastic. Now the next characteristic on the basis of which we are going to divide the whole animal kingdom is segmentation. Then what is the segmentation and what is the difference between the segmentation and symmetry? What is the difference between segmentation and symmetry?
Ma'am, segmentations means the there will be a lines, ma'am, like divisions will present on the body, ma'am.
Okay.
So that is called segments, ma'am.
So in symmetry also we are dividing now.
Ma'am, that's we dividing, ma'am, but it's
a natural division,
yes,
right?
yes,
When naturally the body structure is dividing into small segment. Okay, small parts, small portion. Then this type of arrangement is called as segmentation. Okay, so there are 2 different types of segmentation, whether it is a true segmentation or false segmentation. True segmentation is called as metameric segmentation. Okay, and this type of arrangement is called as metamerism. Arrangement is what? Process is meta. Segmentation is also called as metamerism. metamerism. Okay. So what is metamerism? Metamerism is a body structure having repeated segments. Okay. And it helps to develop specialization of organs. And the best example is annelids, earthworm. Yeah. So true segmentation means if the body is dividing into different segments internally as well as externally. Okay. Then only we will call it as a true segmentation, true division, true metamerism. Okay. So in the arthropods, the insects, you will find the segmentation externally only, but internally they are not divided. Okay, whereas in the annelids, okay, you can find the organism, the whole body is dividing externally as well as internally. But in core data, for example, humans, externally we are not divided, but internally, yes, we are divided, right? Internally our body is divided. So there are two different types of segmentation, external and internal. Another one is pseudo-metameric, that is false segmentation, means when the body get developed, the zygote get, one second, huh? When the organism is getting developed from zygote to the whole organism. So during this development, during the embryonic development, the body is not the body is not having the body is not having segments. Okay, but after that, after the complete development, they will start showing segment. This type of segmentation is called as false segmentation. Clear? And the example is tapeworm. During the embryonic stage, you will not find the segments. Okay, but once they get developed, mature, okay, adult one, they will have segments. Clear? Aarti, let's take a small break for 5 minutes and then we will continue. Okay? Hello. Hello, Auntie.
Yes, ma'am.
So now hello, Auntie. Am I audible? Yes. So our next characteristic on the basis of which we can divide the whole animal kingdom is body coelom or cavity. Remember, I told you that germ cells differentiate into three layers. The external ectoderm, next mesoderm, next one is endoderm, right? And this one, this one, this inner thing what, okay, is what is the mass blastophore, right? So this mesoderm, okay, if the mesoderm is forming the body cavity, okay, the cavity, if they are making the cavity, then only we will consider it as a actual body cavity or body coelom. So what is coelom? What is cavity? This coelom is the name we use to give the internal body cavity that separates the digestive tract from the outer body wall. Okay, so outer body wall, the skin, the dermis is derived from muscles, is derived from ectoderm. Okay, and if you will go inside the ectoderm, you will find mesoderm. So this mesoderm cells are deriving the cavity, body cavity. Okay, so this internal body cavity, which means coelom is derived from the mesoderm. Now the endoderm will form visceral organs. Okay, so between your organ and the muscle layer, between your organ and your external body wall, there is having space, right? So this space, this cavity is what? Coelom. Okay, and this space, this cavity is filled with a fluid which is called as coelomic fluid. Coelomic fluid. Okay, and this fluid is lined or covered by the layer is called as peritoneum. Peritoneum lined or covered what? Coelomic fluid. Okay, which means this peritoneum is derived from mesoderm only, right? Very. Now it is clear what is coelom. On the basis of the nature of coelom, the organisms are differentiated into three types. If the organism is not having the cavity, the coelom, okay, so absence of coelom, here the A represents absence. If the coelom body cavity is not there, then such type of organism is called as acoelomate. Okay, and the examples are Platyhelminthes, Porifera, Coelenterata, and Ctenophora. Why we are not considering Porifera? Sorry, yes, we are considering the Porifera. Is it clear? Okay, now I am going to show you one image that will help you to understand body cavity or the coelom. Just few minutes back, we had discussed about the germ layer, right? Diploblastic and triploblastic. Yes. So all the organisms who does not belong to the triploblastic, then that which means all these organisms definitely they are not having mesoderm, and that's why they are not going to have a coelom, right? So this is the coelom. As you can see in the image, this is the ectoderm. This one is the endoderm. This is the mesoderm. Mesoderm forms the cavity, and they lined single layer of cell, and this line was covering the cavity, the space, okay, the fluid is peritoneum. Okay, clear? So this is the coelom. Okay, if it is not there, as you can see here in the image, it is not there, right? This type of space are not there. The whole body is having layers of cells, right? And then inner body mass, right? So this is what? This is acoelomate. Okay. Next is pseudocoelomate. So look at here. This is the ectoderm, endoderm, and the mesoderm is trying to make a cavity, but they are not able to make a proper body cavity, and that's why they make a pouches of that, okay, and they scattered in their space, okay, the space where the mesoderm is actually present. In that particular area, they will be there, but in a scattered and pouches form. So they are showing that they are forming coelom, but this is actually not a coelom, not a body cavity. And that's why it is called as false coelom, false body cavity, or pseudocoelom. Here, what is pseudocoelom? And the example is only and only nematodes, Escalmenthes. Clear?
Yes, ma'am.
Oh.
Ma'am, in A coelomates, mesoderm also absent, ma'am.
In acoelomate, mesoderm will be yes, definitely. Mesoderm will be not there. That's why they are not able to make a coelom, because mesoderm will get derived from the, sorry, coelom will get derived from the mesoderm only. Okay,
Yes,
and
ma'am.
that's why it is porifera. Sorry, that's why it is porifera, Coelenterata, Ctenophora, because they all are either no germ layer or diploblastic. Okay,
Yes.
triploblastic, they will start showing the coelom, but first they will show the pseudocoelom. The first organism who is having triploblastic, they will have a pseudocoelom. Then they are getting more evolved, and then they will have a proper coelom. So pseudocoelom is clear to you.
Yes, ma'am.
Example, only one example we are having, that is nematodes, escarpment. Next is coelomate. If the body is having true cavity, which is derived from the mesoderm only, then we can say that yes, that particular organism is true coelomate or coelomate. So for the word true coelomate, we can use eucoelomate. Eu means true. Okay. So we are having three types of coelomate. One is schizocoelom. Schizocoelom means when the mesoderm, okay, when the mesoderm layer will split and then make a cavity. And the example is arthropods, mollusca, and annelida. Here the mesoderm layer cells, they are not producing, but the cell layer is itself getting split and then making the cavity. Next is enterocoelom. When the mesoderm layer cells are forming the wall, that is peritoneum layer. Okay, the wall. Mesoderm is making a peritoneum. Okay, and mesoderm is itself getting derived from the wall of the embryonic gut. Okay, mesoderm will derive peritoneum. Yes, we had already discussed about that. And here enterocoelom, this mesoderm, okay, this mesoderm will this mesoderm will derive the peritoneum, and then it will make a embryonic gut. Okay, so during the formation of embryonic gut, what will happen? They make a hollow outgrowth. This is the hollow outgrowth. And then after some time, what will happen? The ectoderm. Hello.
Yes, ma'am.
Hello.
Can you hear me, ma'am?
Yes. So this is the wrong label. This is not the ectoderm. This one is the ectoderm. Okay. After the formation of this hollow outgrowth, the ectoderm from this absent part, they will make a uniform layer. Okay. Now this mesoderm will make a proper structure, that is coelom, and internal layer of the coelom will make embryonic gut. Here,
Yes, ma'am.
so this inner layer will make a gut. Okay. Now this is the peritoneum. This is There are the n numbers of layers of cells, which is actually a part of the mesoderm. Okay, and they will make proper cavity. So this is the developing cavity. So once they both will come together and they will merge together, then only it will be the complete coelom. Before that, this is the developing coelom. Is it clear to you how the intercoelom will get formed? We are having external layer, ectoderm, right? So during in an enterocoelom, what will happen? This ectoderm first, what will happen? It will split. Okay. Now from here, they will start making the inner layer. They will start moving innerward. Okay. Ectoderm. There are mesoderm. Okay, and this one is the endoderm. Okay, after that, this part will get detached from the ectoderm. Okay, and now the ectoderm will make a uniform layer to pack the structure. Are you getting me? No. Yes. Okay. Now we are having structure like this: ectoderm. Internally, we are having structure endoderm, and this layer, the inner cell mass. Okay, this inner cell mass will form a gut. What happened to this layer? The mesoderm. This mesoderm will make a small pouches. Okay, both the side of the endoderm. After that, these pouches will start increasing their size. Okay, by that time they are keep on increasing, when they will, both the ends of both the pouches will submerge and make a uniform layer. Okay, like this. This is the endoderm. This. Yes, and this is your outer ectoderm. So this is what? Coelom. Okay. Before the submerging of these two ends, it is called as developing coelom. Clear? Enterocoelom. This enterocoelom you can find in echinoderms and chordates. Echinoderms means starfish. Okay. Next is hemocoelom. This hemocoelom means the body cavity will be small in size and it will be filled with blood. So in insects, we don't use the word blood, but we use the term hemocoel, right? Right? Hemolymph. So this blood which is present in a coelom, reduced coelom, is called as hemocoel. Clear? Now the next basis of classification is notochord. What is notochord? What is notochord?
It's a rod-like structure, ma'am.
Is it? Notochord is rod-like structure. Your bone is also a rod-like structure. So can we call it notochord? Yes, Aarti. In the last class only you said that you already did animal kingdom. Which cell?
Mm. Ma'am, it's formed only on the uh, embryonic development, ma'am. The structure.
Hmm. So this notochord is a rod-like, but it is made up of cartilage. Okay, so cartilaginous rod-like structure which is present in the body of embryo or which is present in embryonic stage plus in some of the adults also. Okay, so here you can see the cartilaginous rod-like structure that supports the body, right? That supports the embryo basically. Okay, so this is what? This is notochord. Okay, as it is a made up of cartilage, so it is solid, unjointed means it will be the uniform rod. There will be no joints, no branching like this. Okay, the uniform single rod will be there that will support the body and it is made up of cartilage. Clear? So the presence or absence of this notochord in the animal, on the basis of that we can divide the animal into two groups. So one is non-chordate. Non-chordate are those which does not having notochord, like porifera to hemichordates. All the lower organisms, the sponges, annelids, arthropods, coelenterata, ctenophora, platyhelminthes, till the hemichordates, they all are called as non-chordate because they are not having notochord, which means they are not having cartilaginous body supporting structure. Okay, and all those organisms who possess notochord, they are called as chordates, means they are having notochord either in the embryonic stage or it will be keep on continue in the adult stage also. Clear? What is notochord? Now the next basis of classification is types of eggs. Okay, so we are having different types of eggs: microlecital, mesolecital, macrolecital. What is this micro, meso? Any idea? Types of eggs are the
Ma'am, I think they are categorized by the size.
Size of what? Hmm.
Size of the cell.
Size of the cell. You said that you already studied animal kingdom. Hmm. Yes, asking.
Types of the egg is not there.
Types of eggs are not there. Okay.
No.
So you already know what is egg, right?
Yes, ma'am.
The outer proteinaceous layer. Are you vegetarian or non-vegetarian? First tell me that.
Non-vegetarian.
Non-vegetarian, then definitely you know what is it. So the outer white layer is completely made up of protein.
Protein.
Okay. Inside the white layer, you can find the yellow yolk, right? This yellow yolk is nothing but the zygote, and now it will undergo the process of cleavage and it will develop into the embryo, right? So, on the basis of quantity of yolk, okay, on the basis of quantity of yolk, write it down. Quantity of yolk. We are having different types. Micro, lecital. Micro means if the content, if the quantity, amount of yolk is very, very, very small, like in a sea urchin, okay, like in a herdmania. They are having a very small amount of yolk. So they are called as microlecital. Small amount of yolk. Mesolecital. Mesolecital means, now you can tell me. Mesolecital, if the egg is having moderate amount of yolk. Okay? Moderate amount of, okay? So, mesolecital. And the examples are frog, toads, lamprey. Please write it down, the example. Examples are very much important in this chapter, okay? Examples of mesolecital eggs. Lamprey, frog, toads. And then next type is macrolecital. Means the egg will have large amount of yolk. Okay? Large amount of yolk. As you can see here in the image also. The content amount of yolk. This one is representing yolk. Okay? So this macrolecital is further divided into homolecital, telolecital, neolecital, centrolecital. Okay? So this neolecital, centrolecital, homolecital, or this type of classification is based on the distribution of yolk. This division of uh, the division of macrolecital is based on distribution of yolk. Based on distribution. Please write it down, distribution of yolk. Which classification? Macrolecital. Classification or categorization of macrolecital is based on distribution of yolk. So first one is isoleucithal or homoleucithal. Homoleucithal or isoleucithal. If yolk is uniformly distributed throughout the egg, okay, then it will be called as homoleucithal. Example is eggs of Annelida, mollusca, echinoderm, protochordus. Iso or homoleucithal, uniform distribution. Examples, did you wrote examples of homo or isoleucithal? Annelida, Echinodermata, yes, mollusca. Next one is taloleucithal. Okay, it should be E, not A. Teloleucithal. Teloleucithal means if the yolk is concentrated in a vegetal half. As you can see here, vegetal half means more than half. Okay, if the yolk is concentrated in a vegetal half, vegetal half, concentrated in a vegetal half. Example is eggs of amphibians. Eggs of amphibians. Centroleucithal, as the name itself suggesting, if the yolk is localized at the center. And the example is insects. Eggs of insects. Please write it down. Eggs of insects. Understand? Meoleucithal. Meoleucithal means the yolk is very large, that much large that it is occupying nearly entire cytoplasm of the egg or ooplasm, okay, and leaving a very small space, very small area, okay, which is looking like a disc, okay, and that's why it is called as neoleucithal. You can see leaving a very small area like a disc, and so it is also called as discoidal. Discoidal or neoleucithal is the same. Discoidal. Discoidal and neoleucithal is the same. Examples are eggs of reptiles, birds, and egg-laying mammals. Do you know the name of egg-laying mammals? In platypus. Platypus is the platypus. Platypus is the egg-laying mammal. Okay. Is it clear to you? Types of eggs. Okay. Now the next basis of classification is cephalization. Cephalization. Diencephalon. Remember? Cephalization. Diencephalon. I think this term is quite common, and I'm 100% sure that you must have heard this word diencephalon. Cephalon. Do you? No.
Yes, ma'am.
Yes or no?
Yes, ma'am.
What is diencephalon? Right? Diencephalon means two parts, right? So,
Brain,
cephalize,
ma'am.
yes, means cephalization is related to your head. Okay? So, cephalization is the differentiation of head at the interior end. Okay? When there will be a differentiation of a head at the interior end, and it will form the structure. This is called cephalization. So the inside the head, if uh, you know that inside the head there are nerve cells, there are ganglia, right?
Yes, ma'am.
So what happens in a cephalization process? These structure, the nerve cells, ganglia, they are start collecting at one point, and that is inside the head, at one part of the body, at the different part of the body, that is head. Okay, so this collection, aggregation at one point inside the head, this process is called as cephalization. Okay, and this type of formation, aggregation, clustering of the nerve cells at a different position, at a different place in the body, that is head, this begins from the platyhelminthes. So you can see the cephalization process starts with the platyhelminthes. Before the platyhelminthes, Ctenophora, Coelenterata, Porifera, you will not find this formation. Okay, so
Yes, ma'am.
even we can say that the cephalization is the basic, or from the cephalization we can conclude that the organism is getting evolved, because whenever we talk about the evolution, our main focus is to the development or evolution of brain or the brain cells. Clear? Now, the next basis is appendages. What is appendages? Appendages is the body parts that are projecting in such a way that it will help in the process of locomotion, movement. And why locomotion and movement is necessary? For the protection, for the feeding, right? So, appendages is one of the characteristics on the basis of which we can divide the animal kingdom. Okay, so different organisms having different appendages, like for hydra, that is the example of coelenterates. The name of appendages is tentacle. They use tentacles for sensation, to catch the prey, means for the feeding. Earthworm, nereis, basically annelids, right? They use parapodia. Parapodia means, if you can see, they are having segments, right? So first they will uh, relax the segment, and they will contract, then relax, then contract. Each segment will contract and relax, and by this process they will move further. This type of movement, where the contraction and relaxation of segments are going on, is called as parapodia. So here the segments are the parapodia, okay, which is helping in the movement. Then foot or the muscular. In the mollusca, they use muscular fit. If you can remember the structure of octopus, they have a n numbers of tentacles. Why we are using tentacles in octopus? Because the tentacles look the similar what you will find in Coelenterata, the same muscular structure, but this is muscular, whereas in Coelenterata the tentacles are not muscular. Okay, then antenna and legs. Yes, in cockroaches or any insects, you must have seen the head part is having two appendages, which is called antenna. This antenna is just for the sensation. Okay, other appendages throughout the body, they use as a locomotory appendages, they use for to catch the prey. Okay, but the antenna is just for the sensation or the signal. Then starfish, Echinodermata, they have a tube fit. Fishes, they are having fins. And from the amphibians to the mammals, either they will have a wings or the limbs. Okay, so this is all about the appendages. Now let's see some questions. Which one of the following group of animals is bilaterally symmetrical and triploblastic? Yes, Archie.
Yes, ma'am.
Answer it.
Sponges, ma'am.
Sponges. Sponges are having cellular level of organization. Then how it could be triploblastic? Triploblastic means three germ layer.
Three germ layers. Yes, ma'am.
And bilateral also. So definitely sponges will not be the answer. Okay. Now,
Yeah, ma'am.
mid area, Coelenterata. Huh? They are having radial symmetry because this is the sessile. At one place they are fixed and they have to catch the prey. And are you getting me? So how they will move? They cannot move, na? So they will use their tentacles, and that's why they are having radial symmetry. So this is the info. Now, remember, triploblastic is much evolved, okay? And these
Roundworms, ma'am.
exactly, yes, round. Okay, if the live earthworm is pricked with the needle on its outer surface without damaging the gut, this is the outer layer, okay, inner layer, endoderm, and this is the gut. Which one is gut? Let's write it G. This is the gut, okay, and you are pricking the layer, okay, and here the question is you are pricking the outer surface, okay, without damaging this gut, okay. The fluid that comes out is, which fluid will come out?
Slimy mucus, ma'am.
Slimy mucus, is it? If you will prick the outer surface of the body, whole body, they are asking about the whole body, means you are pricking the whole body.
Make a fluid, ma'am.
Which coelomic fluid, very good. Which one of the following is not a characteristic of phylum Annelida? Closed circulatory, segmentation, pseudocoelom, ventral nerve cord. What will be the answer? Do they have a segmentation? Annelida, earthworm. Do they have a segment?
Yes, ma'am.
Yes. So this one will not be the answer. Hmm. Do they have a circulatory system? Either the open or closed.
Ventral nerve cord, ma'am.
Ventral nerve cord. Annelida is a chordate or non-chordate? First tell me that. Annelida is a chordate or non-chordate?
Chordate, ma'am.
Chordate, sure. From porifera till hemichordata, they all are non-chordates. Okay? So this is Again, not a characteristic of annelida. So it is a characteristic of annelida because they are having a reduced form of ventral nerve. Okay, not a proper ventral nerve. They having reduced, but still they are having. So we cannot consider the answer for this question. Hmm.
Closed circulatory system, ma'am.
Closed circulatory system. Do they have a circulatory system?
Ma'am, they have segments, ma'am.
Please read the question properly. Which one of the following is not a characteristic of annelida? Not. Till now, whatever characteristic we have studied, please revise that in your mind and then answer. Do we study about circulatory system till now? No. I only answered your doubt, okay? But we didn't discuss as a topic. What is pseudocoelom? I told you pseudocoelom,
False coelom.
false coelom, and you will find this coelom, pseudocoelom, only and only in nematodes, not other organisms. So which one is not a characteristic of annelida? Pseudo,
Pseudocoelom.
yes. Ascaris is characterized by. Ascaris is what? What is Ascaris?
Don't know, ma'am.
Ascariasis. Yes, it is a worm. Okay, it is an endoparasitic worm, and it belongs to uh, nematodes, Escalminthus. Okay, so Ascaris or the nematodes means they are having true coelom, false coelom, no coelom. What will be the answer? Do the Escalminthus are having coelom? Do they have coelom, true coelom?
False coelom, ma'am.
False coelom, which means presence of neither true coelom nor metamerism. Okay, so the correct answer will be option D. So in today's class, we had discussed about the basis of classification. How can we classify the whole animal kingdom into different groups? What we will study on the basis of what we can divide. We studied here about the level of organization, cellular level, tissue level, organ system level, right? Then body cavity, coelom, whether it is present or not, symmetry, the radial, biradial, bilateral, yes. Then appendages, yeah. Then diploblastic, triploblastic. These are the basis on which we can divide the whole animal kingdom. Okay, so any doubt in any concept? Animal kingdom, most of the students are having n numbers of doubts. Yeah. So what you will do after each class, revise and make a note of your doubts.